discuss the rest of the clinical trial. The first one, I will talk about the data. What are the types of the data? So we have two types of the data: categorical data and numeric data. So first, what is the categorical data? Categorical data subdivided into two types: nominal and ordinary. Nominal, that means the answer it will be yes, no. So the data it will be like yes or no. But the ordinary, it's a gradient, like we said, mild, moderate, and severe. Numeric data, that means they are numbers. So the types of the data subdivided into categorical data and numerical data, and the categorical data subdivided into nominal and ordinary. Then we talk about the data flow. So the data flow, the first one is a study protocol, and then we make the data collection. Who is the collect the data? The investigator. So we start with the study protocol, data collection, then the source data, which is the health record, then the data capturing, then we capture the data in electronic data capturing, which is abbreviated as EDC, or electronic case report forms. We see E, that's mean electronic, C, case, R, report, and F, forms. So then the data analysis and reporting, and it will be end with a clinical study report. So this is the data flow. So we start with the study protocol, then source data, then electronic data capturing, then clinical study report. So we have sponsor and investigator. Sponsor, that is responsible for the trial setup. So the the jobs, or we can say the tasks, like the study design and the database space, all they are responsibility of the sponsors, which they are related to the start, or we can say the trial setup. So the study design on the database. Then it's come the conduct. The conduct, we have the data collection and form the source data by physician and patient, and that is the responsibility of the investigator. And the investigator is responsible for entering the data and capturing them in electronic data capturing. Okay, then we go back to the sponsors, which is responsible for the data validation and the clean up. So the sponsors responsible for the data validation and the clean up, then responsible for the database lock, data analysis, and data reporting. Then we talk about the source documents. The source documents may be original documents, data, and records. Responsibility of the investigator, we said alowaka, alocoa, and we said that attributable LCOA, and we discussed it before. Then we talk about the patient reported outcome. P, patient, R, reported, O, outcome. Provides better understanding beyond data, like health related quality of life, symptoms, functioning, stratification, decision making, preferences, and the treatment compliance. So first, I will talk about the health related quality of life, which is a part of the patient reported outcome. The health related quality of the life, which is abbreviated as HR, that's mean health related, Q, that's mean equality, O, that's mean of and life, may be generic or specific. So we have like types of these. So EQ 5D, which mobility, self-care, usual activities, pain discomfort, anxiety and depression. QWPSA, mobility, physical activity, social activity, symptoms. SF6D, which is from a questionnaire that is asked about the physical function, rule limitation, social function, pain, mental health, vitality. And HALEX, self-rated health, physical activity limitation. HU12, sensation, mobility, emotion, conjugation, self-care, pain. HU13, vision, hearing, speech, ambulation, dexterity, emotion, cognition, self-care, and pain. And this is Wong Packard faces pain scale that they uses, like 0, that's mean no hertz, 2 hertz a little bit, 4 hertz a little more, 6 hertz even more, 8 hertz a whole lot, and 10 hertz worst. Then measuring PRO, that's mean related outcomes. It can be written, it can maybe interview, face to face or via phone, or electronic patient related reports. I mean, outcome. We can say related outcome. Electronic data capturing clinical trial data is collected into an electronic form, like all the stuff that is related to the patient. Case report form contains all the protocol required information from each subject that need to be reported to the sponsors. Electronic case report form available. And then the central data review. Data management checks database for obviously wrong data. For example, if it's the age is 170 years old, there is no human in that, and missing data or outliers or inconsistent data, sending automated data queries to the site, review data point X. And that is the responsibility of the sponsors. Also, the database lock prevents further changes to the database done after review and the query resolution. Database ready for analysis. Final database lock activities add data that would unblind the study, merge randomization code to assign treatment groups, flag database record as locked, deliver database to biostatics and the clinical pharmacokinetics. We can say it's a SAS datasets, and data reconciliations, ECR and IXRS. We said IXR, that's mean interactive web and voice response system. Then we talk about the clinical study report, CSR. Study description containing study protocol, investigator brochure, competing products and other treatment for the same disease, instructions manual, statistical analysis plan, randomization and blinding schemes, tables listing and figures, data monitoring commits, minutes and recommendation, disease process, and then the structure or the structure of it. The first is introduction, methods, study subjects, study populations, like intention to treat population against the protocol, efficacy, safety, and conclusion. In term analysis, data analysis before data collection is completed. Sponsors, as we said, the study design is responsibility of sponsors, database is responsibility of him, and the data validation and the cleanup. So it goes in term data monitoring, commit, unblinded, and then this responsibility of the sponsors. Again, we have the study design, the database build, clinical leader, data management, they do the protocol, then the investigators, then monitor the data and do all these stuff. And I said all these functions are the responsibilities of the sponsors. The database infrastructure, so A is the protocol, the trial must, like this is the database infrastructure, the database analysis of the clinical data, external data, like collection of clinical data, electronic clinical record forms, and also IXRS, drug supply and pharmacological trial management system, trial master file, and then the site, the patient, the clinical data, and the main thing is the protocol. Then we talk about the clinical data management, the task collection, integration, validation of a clinical trial, the functions involved in all the aspects of the data processing software, database, cleaning and management of the data. And here we see this stuff like protocol, feasibility, BIC protocol unsigned, and we said that before. Principles of a clinical data management. Database contains all data required to document safety and efficacy. Database matches data filled in a central records, data cleaning, and complying with the relevant laws, regulation, and guidelines. Analysis sets defines which patient population is included in the analysis, disposition of enrolled patients, summarize protocol violations, degrees of compliance and missing data effects, analysis sets, reason, minimize bias and demonstration of lack of sensitivity, Full analysis sets all the patients that receive the first treatment, intention to treat. Pre-protocol set all the patients who receive the full treatment. So we have two types of the trial: suppository trial and non-inferiority trial. Suppository trial, overly optimistic results, and non-inferiority trial, conservative results. About the missing data, it may be problematic, bias, decreased power, type 1 error, false positives, and last observation carrier forward. Last observed score is used for all the subsequent observation points. Overestimation of efficacy or underestimation of the safety issues. Baseline observation carried forward, used when endpoint is expected to return to the baseline value after withdrawal. Using representative value subject to treatment average. Mixed models repeated measured. Estimated treatment effect via extrapolating previous subject specific effects and correlation between the previous treatment. Standard multiple imputation. Consider uncertainty about the missing data. Several different plausible data sets are created. Like we have many types of the missing data, like problematic, last observation carried forward, baseline observation carried forward, mixed models, repeated measures, standard multiple imputations. And then we have something is called Kaplan-Meier plot, moves all the patient to the same baseline. Like this is a drug A, the drug B, and we try like we move them in the same baseline. And then the investigational product. What is the investigational product? Pharmaceutical form of an active ingredient or a placebo that used as a reference. So it's a pharmacological something that is composed maybe of the active ingredient or a placebo that is used as a reference, and maybe an already approved product, maybe approved, maybe non-approved. Okay, available non-clinical and clinical information on investigational product must be adequate to support the proposed clinical trial. Drug substance, active pharmaceutical ingredient, drug product, finished dosage form. That is the difference, because the drug is the active, only the active ingredient, but the drug product is finished dosage form, maybe tablet, maybe capsule, and it's composed of three things like active pharmaceutical ingredient, excipients, and impurities. Excipients, everything in the dosage from except the drug substance, or we can say inactive pharmaceutical ingredient, but impurities like unwanted components in a new drug product. Investigational a new drug, investigational medicinal product maybe can be already proved the product. IM dossier. IM dossier, like a full document about all the information about this drug. And there is something is called comparator product, is investigational or marketed product used as a reference in a clinical trial. It may be active control or a placebo, like a reference. Okay, and then a pharmaceutical formulation combining API with the excipients together, we can say it's a pharmaceutical formulation. Galenic formulation, optimization of a drug according to the stability and solubility. Stability, degradation, physical stability, microbial stability, like this stuff. Okay, and here we have the galenic. These are the things how to optimize the drug. That's mean how to make the galenic formulation, and that is depend on the efficacious pharmacokinetics, pharmacodynamic application from the patient adherence, manufacturing, robust process, cost effective, stable, like shelf life, transport, storage, differentiated product. The drivers like molecule specific delivery needs, clinical advantage, patient compliance, intellectual property. The routes of the administration: oral gut, topical, percutaneous, transdermal through the skin, intravenous plasma, intramuscular muscle, intrathecal, cerebrospinal fluid, ocular eye, inhalation lung, rectal gut, vaginal vagina. Bioavailability, pharmacokinetics and pharmacodynamics. Studied drug accountability record keeping, drug accountability records, and assurance that the drug is managed to correct the subject at assigned doses. Key part of regular monitoring visits. preparation, dispensation, inventory, transfer, distribution, return to site, return to sponsor, on-site disposal. And the study drug manufacture, good manufacturing practice include materials, production, packaging, repackaging, labeling, quality control, release, storage, and distribution. And then we talk about the study, drug packaging and shipment, plating maintained in preparation and packaging, controlled shipment according to temperature, humidity, and other stuff things. Study drug unblinding only done if the treatment depends on the knowledge which agents was administered. And who do that? The global manager or the investigator. The study drug storage detailed in a structure regarding the storage, including in the shipment and the protocol, continuous tracking of storage condition like temperature log. Challenges and risk of IMP, and we said IMP. Okay. The first things manufacture is more complex, like packaging, no routine production, often one batch per formula, increased risk of mix up and cross contamination when done by hand, delicate supply chain prone to distribution. And about the labeling, primary package in contact with the drugs, secondary packaging, cardboard, name, address, telephone number of the sponsor or investigator, pharmaceutical dosage form, like what are the contents of labeling, route of the administration, quantity of the dosage unit, patch and code number to identify the contents, trial reference code, trial subject identification number, name of the investigator, directions of you. And for a clinical trial, use only, storage condition, expiry date, and keep out of reach of children. Temperature component of the IMP. First one is baby critical for stability, control during transport and storage. Short-term possible deviation may be allowed the shipment without temperature logger. Guaranteeing of a drug is temperature execution accord. Sponsor decides whether to use the affected study medication or not. Reasons for drug supply delays: not enough animal toxicology data, manufacturing issues like impurities or the active pharmaceutical interagent not available, or stability issues, or sometimes is a custom delay. Then we talk about the FDA approval, investigational new product, a new drug, and a new drug applications. Like this is the preclinical research, this is the preclinical, and then the clinical studies, phase 1, phase 2, phase 3. This is the investigational new drug submitted. It's between the preclinical and the clinical studies, and then the FDA review, like new drug application submitted, review decision again, and then this is the advisory commit. Then we talk about the IMP handling. And all these are responsibility of the sponsor's pharmacy, approval of a clinical trial, receipt and recording of safe delivery of IMP, safe handling and the storage, temperature monitoring, report of the deviation, risk assessment of storage areas outside the pharmacy, quarantine, relabeling of expiry date, unblinding, drug accountability, return and disposal of unused IMP, reconciliations of IMP amount expected equal to amount returned, drug alerts and recalls of IMP, maintain pharmacy study file, training of a clinical trial pharmacy staff, archiving of a clinical trial documentation. Okay. And then we talk about the aspects of understanding a clinical trial, approval of the pharmaceutical and medical devices, research study, development study, strategy, pricing and reimbursement, medical information, business decision, marketing claims and sales, treatment decisions and guidelines. And here, evidence-based medicine, gathering medical information, evaluate its quality, make medical decisions using the best evidence. These are the filtered information, these are unfiltered information. The filtered information, systematic review, critically appraised topics like evidence synthesis and guidelines, critically appraised individual articles, and unfiltered like randomized control trials, cohort studies, case control studies, reports and background information. Now we go to the systematic reviews, gathers all relevant studies on the specific study. And then now we talk about the meta-analysis. Use statistical methods to summarize the results of the gathered studies, clinical practice guidelines, like informal suggestions of approachable, optimal, and appropriate care for a specific clinical situation, basis of most clinical decisions, foundation of a clinical teaching, evaluating care patterns and the standards of care, contains methodology and assumption, evidence review, summary of finding, decision making rules of recommendations, rationale for each conclusion and recommendations. And now we talk about the types of it, like single author with expert opinion or systematic literature review, consensus panel, expert optioning, or evidence-based approach. Development of the guidelines, defining clinical questions of insert, summarize evidence on efficacy, dangers, and cost, studies pros and cons, recommendation statements like finding, document all the aspects. Reporting guidelines for the main study types, um, like randomized trials, concert, observational studies, strobe, and systematic review, Prisma, study protocols, Spirit, diagnostic prognostic study start, case reports care, clinical practice guidelines agree, qualitative research SRQR, animal preclinical studies arrive, quality improvement is required, and economic evaluations years. This is the concerted checklist that is composed topic, title abstract, introduction, methods, results, discussion, and other information. And now we talk about the publication bias. So the study with the positive results are more likely to be published, more likely to be submitted, and more likely to be published quickly. Distorted scientific record hides the truth, influence the decision making doctors and policy makers, harm patient and cause type of scientific and research misconduct, like all these the definition types of the publications bias. So we have types of the publication bias, publication or not publication, time lag bias, rapid or delayed publication, multiple publication, finding published more than once, location bias, ease of access, situation or non-situation, language and outcome reporting bias, selecting reporting depending on the results. And finally, the authors responsible and accountable for published work. Substantial contribution to conception or design, acquisition, analysis, or interpretation, data drafting, or revisions. And by this, we end all the clinical trial. Hope you understand thing, and I will maybe like do some mapping or recording to make it easy also. Thank you so much for listening. Hope you understand anything.